National 5 Biology, Multicellular Organisms, video number 2, Reproduction and Variation and Inheritance. If we look at reproduction first of all, reproduction is the process by which the members of a species produce offspring. Sexual reproduction involves the fusion of two gametes or sex cells, for example sperm and egg, during fertilization. In multicellular animals and plants, normal body cells are diploid, but the gametes are haploid. If we have a look at the human female reproductive system first of all, sperm is deposited in the vagina. Uterus, where the baby develops, has a thick muscular wall. The oviduct, which carries eggs from the ovary towards the uterus and where fertilization ideally occurs. In the oviduct, and the ovary, which of course produces the eggs. The human male reproductive system, the bladder, stores urine. Sperm, produced in the testes, is carried to the urethra by the sperm duct. There's the prostate gland. The urethra allows urine and sperm to exit the body, and the penis is what the male uses to deposit sperm into the vagina. And the testes, of course, are where the sperm cells are produced. If we look at the sperm and egg cells themselves, upper right picture shows you how they look to scale. The egg is considerably larger than the sperm. Both are specialized animal cells. The sperm has a tail for swimming towards the egg, as a head containing the nucleus, and the nucleus contains all the genetic information, of course. The egg cell with a cell membrane, nucleus with the genetic information from the female, and cytoplasm containing a significant amount of food for the zygote to develop. If we just run through the various stages involved in fertilization, the egg cell, the follicle containing the egg, develops, and then the egg is released from the ovary. The egg's picked up by the funnel of the oviduct, and it gets moved along the oviduct by cilia from ciliated epithelial cells that line the oviduct. At this point, sperm enter the uterus and swimming towards the oviduct. At this point here, the sperm penetrates the eggs and the gametes fuse, and that is fertilization. The fertilized egg divides a number of times and becomes a ball of cells, which is an embryo. Ideally, that embryo will embed itself in the wall of the uterus and continue to develop. In plants, flowers are the organs for sexual reproduction. The anther and the filament form the stamen. This is the male part of the flower, and the anther produces the pollen grains, which are the male gametes. The stigma, the style, and the ovary form the carpel. This is the female part of the flower. The ovary produces ovules, which contain the egg cells, which are the female gametes. In pollination and fertilization, pollination is when a pollen grain lands on the stigma of a flower. For fertilization to occur, the pollen grows a pollen tube down the style and into the ovary. The male nucleus travels down the pollen tube and fuses with the egg nucleus in the ovule. So to summarize fertilization in animals and plants, the haploid sperm and the haploid egg, each containing a single set of chromosomes, come together at fertilization to form a diploid zygote. The zygote contains genetic information from both parents, and this then develops further to become an embryo. Now let us consider variation and inheritance. Variation is how individuals within and between species vary from one another, and combining genes from two parents contributes to variation within a species. Now, just to recap what genes are, DNA contains many genes. These genes code for proteins, and different proteins determine our characteristic features. These characteristics features can be classified as either discrete forms of variation, which are controlled by a single gene, or continuous variation, which are polygenic, controlled by many genes. In discrete variation, observed values fall into distinct groups, but in continuous variation, the variation itself can be measured. There are no discrete groups, and what can be measured in terms of the variation covers a range of different values. If we have a look at different examples of discrete and continuous variation, for discrete variation, eye color in humans, type of leaf, either variegated or non-variegated, earlobes, either attached or unattached, and fingerprints, for example, either a loop, an arch, a whorl, or compound. Examples of continuous variation, as we said before, covering a range of values, such as height in humans, leaf length in plants, diameter of limpets that you may find on the seashore, and, for example, the length of different petals. Now, in this, it's very important to understand genes and alleles. 23 individual chromosomes in the haploid sperm and haploid egg come together during fertilization to form the diploid zygote. With 23 matching pairs of chromosomes, that makes 46 in total. Now, if we consider one of these pairs of chromosomes, they will each possess the same genes. Remember, one of those chromosomes will come from the female parent and the other one in each matching pair from the male parent. Now let's consider, here's the location of the eye color gene from each parent. These eye color genes from each parent might be slightly different to each other, and we call these variants of a gene alleles. Alleles may be dominant or recessive. If an organism possesses a dominant allele of a gene, it will be expressed instead of recessive alleles. We use uppercase letters to identify dominant alleles and lowercase letters to show recessive alleles. In eye color, example, capital B represents the dominant allele, and this codes for brown eyes. Little b represents the recessive allele, and this will code for blue eyes. If each gene has two alleles, then there are three possible combinations of these alleles. But before that, we must understand a couple of terms. Genotype refers to the alleles of a gene an organism possesses, and phenotype the features produced by a particular genotype. In the genotype big B, the alleles are described as being homozygous dominant, and this codes for brown eyes. The genotype big B, little b, the alleles are heterozygous, and this also codes for brown eyes, and the genotype little b is called homozygous recessive, and this codes for blue eyes. Now, a monohybrid cross shows the inheritance of traits which are controlled by a single gene from parent to offspring, and it's usual to carry out a monohybrid cross up to the second generation of offspring. Something of note, true breeding is the expression we use to either describe a homozygous dominant or a homozygous recessive individual. If these are bred with others the same as themselves, they will always produce the same genotype in the offspring generation after generation. As an example of a monohybrid cross, homozygous dominant or true breeding brown-eyed parent crossed with a homozygous recessive or true breeding blue-eyed parent show the genotypes and phenotypes up to the second generation of offspring. So our original parents, designated with the letter P, homozygous dominant, big B, crossed with little b, are homozygous recessive individual. Now, if you look at the possible combinations of genotypes in the offspring that they can produce from their gametes, they are all the same. The first filial generation of offspring, or the F1 generation, the genotypes will all be heterozygous, big B, little b, and the phenotypes, they will all be brown-eyed. Now, if we take a couple of those individuals from the first generation of offspring and breed them together, so they are our second lot of parents, here's the possible combinations of the gametes as the genotypes of the offspring. in the F2 offspring generation, the genotypes are like so, but the phenotypes are 3 brown-eyed individuals to 1 blue-eyed individual. Now, that particular ratio of dominant to recessive traits is expected in the second generation of offspring when the original parents are true breeding for their trait, brown or blue-eyed. However, it must be remembered, observed ratios of traits in offspring are usually not as you would expect because fertilization is a random process. Now, one aspect that we can use the same type of techniques for monohybrid cross as used for us is for designating genotypes in family trees. If we consider an example, tongue rolling is inherited, is controlled by a single gene, and the tongue rolling allele is dominant over the non-tongue rolling allele. So big T equals the allele for tongue rolling, little t is the allele for non-tongue rolling. So if we use this to try and trace the pattern of inheritance in a family tree, the blue individuals are tongue rolling, the white individuals are non-tongue rolling, circles represent females, squares represent males. So let's consider this family tree as an example. The easiest and first thing to do is identify all the non-tongue rolling individuals. They are the white ones, and of course if they're non-tongue rolling, their genotype will be little t, like so. We can now work out the genotype of the male parent because he has produced individual young which can roll their tongues and also are unable to roll their tongues. So he must be heterozygous because remember, one allele comes from each parent in the offspring. The tongue rollers in the F1 generation must be big T, little t. We can now work out the genotypes of the children in family X. From those two parents, if they're tongue rollers, they must both be big T, little t. Now, before we work out the genotypes of family Y, we can work out family Z. If they have one of their parents which is non-tongue rolling, but all the children in family Z are tongue rolling, they must be all heterozygous. We can't work out the genotypes of the remaining individuals for certain. However, we can have a good guess that the male parent for family Z is probably most likely big T, homozygous dominant, given the genotypes of his offspring. The last individual is impossible to work out. They can either be homozygous dominant or heterozygous for tongue rolling.